Whoa. Now I'm hoping the audio picks up. I see the microphone kind of going up there. I'll edit this later on. Okay, so hope this works. All right, so our topic for this week is nucleic acids. And their importance is essentially to carry and transfer information. Essentially genetic information. And there's a minor role in that some of these molecules also are involved in regulation and also in energy transfer. We're going to see later on. I'm going to focus this unit on the large macromolecules, but the smaller building units that we're going to introduce today, they also play a major role as currency in transferring energy between systems. As usual, we'll start with the building blocks, then we'll build it out into how do you assemble these things into the large molecules, and then we'll look about how that information flows from DNA to RNA to proteins. And then we'll talk a little bit about the control of that information. All right. So let's go ahead and go over here and let me show you a schematic. This is what we call a nucleotide. And this is what we're going to call our, remember we talked about how all these things start with these building blocks, like so monosaccharides are used to build carbohydrates, amino acids are used to build proteins. So this is the building block for nucleic acids. I'm just going to give you a schematic of it. Is that coming up on the board? Yeah. My handwriting is horrible, I'm sorry. I think I just had too much coffee. Anyway, so the point is that what you have is we're going to have a phosphate. That is going to be connected to a pentose sugar. And then that's going to be connected to something we're going to call a nitrogen base. And that's kind of the schematic. Those are the three components of a nucleotide. So let's start looking at the pentose sugars first of all. Let's talk about the sugar. So remember that sugars are compounds that are mainly have a carbonyl and a hydroxyl group, right? So they're kind of like either aldo alcohols or keto alcohols, right? And remember that they have this ability of kind of folding around and forming like a ring kind of structure. And the two that we're going to talk about are called ribose and deoxyribose. Ribose will have a structure that looks like this. Again, remember this is what we call the Hayward's projection. And in this case, I'm going to put them all in the beta conformation. Remember when you make the hemiacetal, remember the carbon once OH ends up looking down, we call it alpha. If it looks up, we call it beta. And again, let's say up or down, it means in this particular projection, which is just a way of showing the molecules. And then the way it works is ribose will have OHs here. And then, of course, we have the CH2 OH over here. Same thing here. CH2 OH. Now the difference is that the deoxyribose is missing a hydroxyl group there. Missing the hydroxyl group. And that's what we call it, deoxyribose. It's missing that OH group in there. So if we want to do fancy with names, remember, we have a name for these things. So we call this guy a beta. This is, again, whenever you see this kind of structure, we know it's the D isomer. Remember, these things have stereoisomers. And this would be ribo, and then 1, 2, 3, 4, 5 sides, furanose. Just to keep us thinking about these things. And then this guy would be, again, beta D. But we had to make an exception here because you could have taken off the OH at this carbon instead of that one, or for that matter, the one over here. So if this is, again, carbon number 1, 2, 3, 4, 5, remember it's a pentose. So this would be 2 deoxyribofuranouse. Furanose means it's a 5-sided ring. Ribo means which is the parent sugar. Deoxy in this case because it's missing an OH. This indicates the position at which it's missing. And then D is the isomer when we look at mirror images. And then beta, because this OH is facing up. And the reason for that is when we look at the overall structure, we'll see that it makes sense. Because remember that I think I showed you, when you make these ring structures in three-dimensional space, the beta anomer tends to point the group outwards away from the ring. And so that's going to be a much better placement when we attach this nitrogen base to it. It's going to look better, and it's going to allow DNA eventually to form the structures that it forms. Now, what's going to happen is the ribose ring is going to be used for what we call ribonucleic acid. Or short for it is RNA. And the deoxy one is going to be used for deoxyribonucleic acid. Forget that now. Nucleic acid called DNA. Now, I want to remind you that whenever you have these kind of structures, some of these centers are reactive. They're susceptible to things. Like, for example, remember that if you join several monosaccharides, you basically join them by making an acetal linkage with this anomeric carbonate. But what if instead of that, we put a nitrogen-based anamine instead of another alcohol? So in that case, you could have, for example, let's say, let's put our ribose here. Yeah, this is OH down, CH2OH. And again, we have the OH looking up here. So let's say that we bring in an amine. NH, and then something connected to it. Well, what you could do is you could form something that looks like this. In other words, you have your pentose in there. Then you have your 2 OHs facing down. And now what happened here was we took these guys here and we liberated some water. But what we have here now is an NHR in there. Now, this is kind of the same thing that we did before with making disaccharides and polysaccharides. But in this case, instead of an O here, what we have is an N. And so because of that, we're going to call this an glycoside. And this is the basis for this portion of the construction here of this nucleotide. Okay? All right. Let me switch this around a little bit so we can take the other part of the board and hope this comes up somehow in your notes. You know what? Maybe I can swing this over here. There. Oh, yeah, I think that'll be a little bit better. Okay, so let's talk about the second component here, these nitrogen bases. And we're gonna see that just like with polypeptides, we have 20 amino acids. We're gonna find that we have an option here of usually 4 bases, but one of those can be switched with another one. So technically it's 5. And this is what we call, these are called heterocyclic amines. And heterocyclic means that, again, you have a cyclic, a ring kind of structure, but unlike the ones we've seen before, not all the positions are carbons. So you have, more than one kind of atom in ring. And we're going to see in this case, what you're going to have is you're going to have rings that are made of carbon and nitrogen. So we have 2 kinds of these. One of them is called purines. The other ones are called pyrimidines. See, this shows up on the board. Yeah, it's going to be a little hard. I'm not going to make you memorize the structures, okay? But there's a very interesting feature. That's all I want you to be able to recognize, okay? Just this feature. This is a structure of a purine. It essentially consists of a 5-sided ring fused to a 6-sided ring. And then it has like alternating double bonds all over the place. This has an H here. So notice where I don't have anything written, just those junctions are just carbons. And of course, when you look at the structure, you have to, assume that wherever there's a bond missing, like this guy here, there must be a hydrogen in there, but we're not showing the hydrogens, except for the one on this nitrogen, because this is the nitrogen that's going to be attaching to the ribose unit. So notice that it has like 2 rings. The pyrimidines, on the other hand, have only 1 ring. And again, you have the alternating double bonds in there. So the difference between these 2, you can tell, right? 2 rings, 1 ring. And all I need you to be able to do is recognize what kind I'm showing. So what's going to happen is you're going to have modifications, different other things attached to those nitrogens in the different bases, but these are the 2 bases for it. Now, the way to remember is the one with the shorter name has the longest or the biggest size. The one with the longer name has the smallest size. Maybe that's a way of remembering them, right? So essentially, all I want you to do is, no need to memorize. No memorizing. Just recognize the 1 versus 2 ring difference. And There's two kinds of purines. One is called adenine, and the other is called guanine. And we abbreviate this as capital A, and this one as capital G. Difference, sorry. There are three possible pyrimidines. You have cytosine, which we abbreviate C, uracil, which we abbreviate U, and thymidine, which we abbreviate T. Well, you know what? I think I misspelled this. Thymidine is thymine. I got ahead of myself. So it's thymine, capital T. Okay, so let's go ahead and assemble the first component here. Remember, we'll have to build this guy with that guy. So let's go ahead and I'm going to erase here, if you don't mind. I would normally write on that side of the board, but I can't get the camera out there, so. Now remember that in the cell, there are all these mechanisms for assembling these things. And as we will talk about later on, there's like one or more enzymes in charge of each one of these processes. So let's go ahead, come back here, and let's go ahead. This is number 3. Let's build what we call a nucleoside. Okay, we're not at the nucleotide level here. This is a nucleoside. So what you're going to have is you're going to have your pentose sugar with an N-glycosidic bond to a nitrogen on the base. So I'm gonna try to do one with this guy here. Right? So this is again our pentose sugar. Forget the OHs are facing down. The anomeric carbon is in the beta form. But remember that this is now going to be an N in here. Okay, so let's say that we do, let's do adenine. Make enough room here. Okay, notice first of all, is this a purine or a pyrimidine? 2 rings, it's a purine, okay? So this one is specifically, this is adenine. And what we do is basically we say, well, if I have adenine plus ribose, I'm going to end up a nucleus with a nucleoside that we're going to call adenosine. If this were the other version, the deoxyribose, then we get something called deoxyadenosine. And one more, let me remind you, you don't have to memorize these structures, I just want you to recognize, like if you look at a picture of it, you're then, okay, that's a nucleoside, or oh, that's a nucleotide, or that's a purine, that's a pyrimidine. Not, you don't have to recognize each one individually. So essentially, with G, for example, we could do the same thing, and we would end up with either guanosine, or sorry, not guanosine, yeah, guanosine, or deoxy, exactly. Very good. One. Now we can do the other ones. Let's say C, U, and T. So C becomes cytidine. Uracil becomes uridine. T becomes thymidine. This is why I made a mistake before I got ahead of myself. Thymidine. And of course, they have their, deoxy versions. Just put deoxy. Okay, so that's what we call a nucleoside. So this is where it starts. So this is only like 2/3 of the building block. That's how you get them. Now, one important thing here is, of course, organic chemists, they like to give things names, and every substituent has a number. So what we do here is we give numbers to the positions on the adenine, and we're going to call this position number 1 here, 2, 3. Oops. Oh, sorry, I skipped one, sorry. So going this guy, this would be number 2, 3, 4, 5, 6. And then this would be position 7, 8, 9. So we would say that in the adenosine nucleoside, the N-glycosidic bond is from carbon 1 of the ribose to nitrogen 9, position 9 on this ring. Now, what do we do with the positions on the sugar itself? Well, what we do is we also give them numbers, but we put a little prime sign on it. So this becomes 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And that's going to have specifically a major role later on, because we'll see that carbon 3 and carbon 5 are the ones that are going to be used to build these polynucleotides later on. Right? So I hope that makes sense. Okay, let's come over here to this side again. And then we erase this portion here. Again, don't forget, you don't have to memorize the actual structures. Just the names, which ones are pyrimidines, which ones are purines. So let's talk about the other component, this so-called phosphate here. So it all starts with an acid called phosphoric acid. I think we've seen this guy before. We said that it's a weak acid and that its conjugates are used to make buffers, for example. So this would be the structure of phosphoric acid. You'll notice something about the phosphorus atom in there, and it is that it exceeds the so-called octet rule. Remember the octet rule we said? We said that atoms typically want to have an entourage of 8 electrons around them, either lone pairs or shared. But for elements in periods 3 and below, because these are big atoms, they can recruit additional electrons and make extra bonds beyond the octet. So sulfur can do that, phosphorus can do that, chlorine can do that, bromine, iodine, these guys, they can do that kind of stuff. Now, remember that we said that if I take an acid and I react it with an alcohol, right? I can essentially do this and form a structure that we're going to call, well, I'll tell you what I'm going to call it in a moment. In the process of doing this, of course, you eliminate the molecule of water. And we're going to call this thing a phospho. You can see here, we're going to call it a phospho monoester. So it's an ester in the sense that it is a product of an acid reacting with an alcohol. We've seen this before, right? It is mono because it's only one place where it added. Notice that the phosphoric acid had 3 possible OHs in that you could have used. And then it's phospho because it's not an ester in the sense that this is not a carboxylic acid. It's a phosphoric acid derivative. Now, we could go ahead and extend this and form something that we could call we put another group over here. We could call this a phosphodiester. Again, notice that there's going to be here 2 esters. That's where the diester comes from, but it's based on phosphoric. Now, I want to remind you that you've seen this before. If R prime is a triglyceride, or actually let's call it a diglyceride, and R is an alcohol. We've seen these before, remember them? These are the so-called phospholipids. And if R prime is sphingosine, and R, again, is an alcohol, this would be a sphingolipid. By the way, you can call this a phosphoglyceride. So these are the phospholipids we saw before. So the phospholipids belong to this category of phosphodiesters. So this could be your glycerol with the 2 fatty acids coming out. This could be like an alcohol or an amino alcohol like serine or ethanolamine or whatever. And that'll be a phosphoglyceride. And if this backbone here is actually, instead of a diglyceride, if it's sphingosine, then we would call it a sphingolipid, and these are the components of membranes. Okay, so now that we have the tools in place, let's go ahead and assemble a full-on nucleotide. Go ahead and do that. Let's see what I have in here. I'm going to try to do this again. Right, I'm going to try to do this guy in the form of a. All right, so here we go. So here we go with H. Let's start over here. H O P O O H O CH2 and here's OH. Here's OH. Here comes again that friend. Adenine. Okay, so see. Two. Oops, sorry. So this is how we're going to call a forming a nucleotide. Again, let me get a different color marker here. So here is our phosphate group. This over here is what we would call a phosphoester bond. Here is our pentose. And here is our nitrogen base. And we said we're going to call this a N-glycosidic bond. N-glycosidic bond. Yeah, notice that the nitrogen base is placed in the beta conformation there. And again, in terms of numbering the positions on in the whole thing, what I'm interested you remember is the positions on the sugar itself. Remember, we're going to call this guy 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So we built a nucleoside, which was called adenosine. Again, it could have been, depending on what this was, it could have been uridine, it could have been cytidine, guanidine, one of those, right? But notice that now we added a phosphate group. So we're going to call this guy adenosine 5 prime monophosphate. Monophosphate because it's only one phosphate group attached. 5 prime because it's connected to the 5 prime carbon of the sugar. Notice that if we don't put that prime, it might confuse us to think that it's connected to one of the positions on the rings here, but nothing gets attached to those rings. Well, later on in modified bases that happens, but as far as the model here is concerned, that's all we need to do. Let's look at some possible variations on this. And many of these exist, and some less common than others. So let's look at some variations here. So of course, one possibility, I'm gonna use some abbreviations here, just to keep it a little easier to draw and stuff, so I don't have to draw everything out there. So I'm gonna replace the full formula here with just letters to represent the bases. So for example, you could have the following setup: CH2OH. And let's say this is, A, same as this guy here. And this is now OH. And now we could have the phosphate over here. So that's a possible variation. And we would call this adenosine. And in the spirit of the numbering system that I just showed you, again, remember this is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So this would be adenosine 3 prime monophosphate. a whole different thing here. Another variation that you may have heard of, probably in your biology courses or physiology or something, is one that has, and again, I'm going to kind of like simplify it here a little bit. HOPOPOPO. Again, also notice that I'm skipping the sorry, I missed one here. EO. You'll notice also that I'm skipping the lone pairs on the oxygens just to kind of keep things simple. CH2. Again, I'm going to pretend this is A over here. Again, this is OH. This is OH. This is kind of a little clunky here. Sorry about that. So you may have heard of this guy. So this would be adenosine 5 prime 1, 2, 3 triphosphate, or ATP. You may have heard of ATP if you've done physiology already. So that's the structure. Again, I'm not drawing the full adenine base in there. But the idea is that this is a molecule that's very important in carrying energy between one chemical reaction and another, and sometimes between distant parts in the cell or even in the organism. And the idea is that, again, we haven't looked at that yet, but the idea is that these chemical bonds with phosphorus and oxygen are very high energy. They're not super stable. In other words, they want to release that energy, and that's why ATP is such a valuable molecule in energy transport. And we'll revisit it. And there are other variations. For example, there is one where the phosphate group is actually connected with both the 3' and the 5', kind of forming like a little ring of itself. And that's called cyclic AMP, and it's a regulatory molecule that influences the action of many different enzymes. So I'm not going to write it here. So this is it's called adenosine cyclic monophosphate, or CAMP. And as you said, this is a regulatory molecule. Sorry, no picture. You know why? Because to be honest, I have a hard time drawing that, sketching that thing out. I mean, you can tell already that I'm not very good at sketching these things, so just bear with me. All right, so let's go ahead and come up with an idea of how these things get put together. Now, I'll probably bring this up. I would probably talk about this in the second lecture, not in this one. But let's try out doing that. So let me come back here and show you. Bring this around here. Yeah, ta-da. So how do you link? This is number 6. Linking nucleotides. Well, there's several interesting things about it. One of them is that the structures that are used to build nucleotides are actually the 5' triphosphates. each nucleotide that gets added are added to the 5' OH of the ribose. And actually, we can say, or I've been doing ribose here. But again, if this were just H, this would be deoxyadenosine 5' instead of deoxy, whatever. Okay. But I'm going to talk mainly about ribonucleotides. What was I going to say here? I forgot what I was going to say here. 5' OH. I totally forgot what I was going to say here. Sorry about that. So let's say, for example, I have this structure here. P O CH2 OH. Okay, so here's my ribose. And let's say this is a base 1. It's either a purine or a pyrimidine, A or G or T or C or U. Well, actually, as we'll see later on, if this is ribose, this is never going to be T. It's always going to be U. If this is deoxyribose, this is never going to be U. It's going to be T. But that's part of our second lecture. Okay, so incoming is another guy. Let's say here base. H, and let's say it's going to be another base here, base 2. Now this guy is coming in, like I said, as a triphosphate. So P, P, H, P. So ready. So where the action is going to happen is right here. Oops, I should have drawn this the other way around. Hold on a second. I need to switch this back on here. I tend to do it the other way because it's easier for me to remember. But this is where the action is going to happen. And notice how you have, again, this is an acid still. That is an alcohol over there. So they can combine to form an ester. So we're going to call this, erase over here. That's good. Do it again. HOCH2. H. And this was base number 1. Base number 1. And then what we have here is we're going to have an OP. This is base number 2. Now we're going to complete this guy here. Notice that we linked it over here. And then what happened was that this guy took off. Now remember, this was 3' over here. And this is 5'. So we would call that a 3', 5', 5 prime phospho, sorry, diester. The other thing that took off is when this guy got chopped off, is something that now has yeah, don't worry which way you run. As long as you know that one of the oxygens is double bonded. This is called inorganic pyrophosphate. But it's such a long word, we sometimes abbreviate it as PPI.
They get separate, so that's the result.
Right, so basically, this guy connects here. So this stays with the growing chain, and then the two phosphates here stay together in this form. It's called pyrophosphate or PPI. This thing over here that we just made, as you can see, we have a 3' 5' phosphodiester. You basically connected them, and so this is what we're going to call a dinucleotide. And as you can imagine, you still have the potential of continuing to add other ones in there and grow this into an oligonucleotide and a polynucleotide, very similar to what we talked about with monosaccharides, oligosaccharides, polysaccharides, or dipeptides, oligopeptides, and polypeptides, kind of thing. So it's kind of saying the same kind of pattern. So the good thing about it is there's a lot of the language that kind of like just stays the same, you just change the ending of it. So it's always, mono, oligo, poly, and you can add in there, saccharide, nucleotide, or peptide to those kind of things. So in our next lecture, we would pick it up here. And we'll go and see, okay, so now let's say that we change this OH into an H. What kind of difference that makes? It makes a world of a difference in terms of, is the molecule destined to store genetic information, or is it destined to transfer the genetic information? That's a big division of labors in there. So let me go ahead and close out here with a little table just to summarize all this stuff that I just did here. See if I can make this have some sense, right? So this is number 7, lucky 7. Let's summarize. Okay, so we're gonna write in here the different kinds of bases that we can have. We said we can have A, G, C, U, or T. So those are some of the bases that we can have. And then we have what's called the nucleotide. Remember, there's always going to be a 5' phosphate attachment. The variation that I showed you, the 3' phosphate, that's not very common. The cyclic format, again, is not used to build polynucleotides, more like a regulatory molecule. But the ones that are used are these guys. And so you can have, we have adenosine monophosphate, diphosphate, triphosphate. And the corresponding deoxynucleotide. The difference being that this would be done with deoxyribose, the one that doesn't have an OH in there. So we simply put a small d in front. So we go DAMP, DADP, DATP. And now if I'm going to be totally correct, it should be ribonucleotide deoxyribonucleotide, but it's just a short form. So again, this would be the same thing. You have GMP, GDP, GTP, and that means how many phosphates are present at that 5' carbon, 1, 2, or 3. And again, over here you have D, GMP, D, GDP, D, GTP. Cytosine, same thing, CNP, CDP, CTP, or DCNP, DCBP, DCTP, uracil, UMP, UDP, UTP, and the deoxy, you don't find it. So the cells do not use uracil when they're building the deoxynucleotides. Similarly, for thymine, the cell does not use thymine to build ribonucleotides. It only uses for the deoxy ones. So TMP, DTP, DTP. Again, I'm not saying they don't exist. I'm just saying the way cells work is this way. So the cells don't use the deoxynucleotides of uracil or the ribonucleotides of thymine. That's pretty much the way it is. Do they exist? Yes, there are. But they're not used for what we're trying to find, which is essentially the carrying and transferring of genetic information. Okay. So again, to notify this, is that U is used for RNA. T is used for DNA. Okay, so I tried to record this lecture and I hope the audio carried. We'll see when I recover the recording lecture. And if it works, thanks for those of you who came in today. And if it works, I'll post it on Canvas by Wednesday or something like that. And you guys can see it. Now remember, Wednesday, we're not going to meet. We're not going to have any meetings on Wednesday. But I am going to post the answers to the worksheet. Maybe if I can get this to work, the recording of this lecture, and a little special humorous philosophy appetizer for this week. I'll put all those in Canvas. Now, today, again, we're not going to meet for lab today, but if you want, we can get together, meet together at 11:30. And I'm thinking that if you guys want to work on the activity, I can make copies of it, but there's a video involved in it. And I don't know, you might want to, if you guys want, I can ask to see if we can go to the Steno instead. If you guys want to stick around and do this thing in the Steno where they have computers that you can use in there and access the videos. Okay. And I will make some copies of the activity that I posted on Canvas, but there's a video attached to it. So this is just the handout. And you have the option of either turning it in on paper or you can also upload it and submit it in Canvas. Don't forget, our quiz on Wednesday is going to be online. You have all day on Wednesday. Any time of the day you can take it. You'll have 20 minutes. It's 10 questions, like always, 10 multiple choice questions. It's all about amino acids and peptides and enzymes and that kind of stuff. And just make sure that you're aware that as soon as you start it, there's going to be a time limit. Now, I think if you are registered for accommodations, you'll get an extension on it. But if not, you'll have 20 minutes is the time. And you have 2 attempts. So I got to check and make sure that it's working. I'm going to check it out today. If something's glitchy, I'll let you guys know. But as far as I can tell right now, it should be working fine. Okay. Okay. Any questions? Hey, thanks for coming and joining us. I hope you enjoy your day today. And like I said, I'm going to check with the Steno and see if it's okay for you guys to park out there. And I'll sit around also in case you guys want to watch the video there and work on this activity. In the meantime, I'm going to make some copies of it. All right. Okay. That's all I got for today. Okay, now how do I stop this? Oh, here we go. I think this is the way it stops, right? No? Oh, here we go.